Hey guys, here to talk about proteins today. We're just going to introduce them, probably get through their structure and everything, because AP does ask a lot about the structure of proteins and then what they do and how they do it. So we need to talk about all that, but we're also going to go into enzyme function. That'll probably be the next lecture. I'll probably split this PowerPoint into two smaller lectures. But let's start with these proteins. So first of all, what do proteins do? And the answer to that is yes, all of it. They do everything. When you are doing something in your body, it's proteins do it. When you think a thought, when you digest an apple, when you move your fingers, whatever you're doing, when you're charging ATP in your mitochondria, when you're flexing a muscle, when you're fighting off a bacterium, whatever it is you're doing, it's all proteins that are doing that, every bit of it. Some proteins you've probably heard of, hemoglobin. What else have you heard of? Hemoglobin. Proteins are chains of amino acids. So the monomer is amino acids. And a polymer is called a polypeptide. And that's because the bond that we form here, right, this bond is a peptide bond. And poly just means many, so many peptide bonds. That's a chain of amino acids. And then that is going to be folded up and sometimes joined with other polypeptides to make the final protein. So polypeptide and protein aren't exactly the same thing. So protein is made up of polypeptides. Sometimes just a single one, sometimes a bunch of them. Hemoglobin, for example, has four different, you can see 1, 2, 3, 4 different polypeptide chains. Rubisco here has got a whole bunch of polypeptide chains. And if you haven't heard of Rubisco, you will, because it's the most important enzyme on Earth. It may look like somebody tried to make a soccer ball out of old chewing gum, but it is the most important enzyme on Earth, and you should thank Rubisco. You should probably thank your hemoglobin too. Actually, take a minute to do that. Just say, thanks, hemoglobin. Hemoglobin's like, she noticed. Anyway, an amino acid has a central carbon with a hydrogen stuck on it, and then 3 other places, of course, because carbon makes 4 bonds, as you well know by now. One of those bonds has an amine group, an NH2, which will sometimes ionize to NH3. It does act as a base. We talked about that before. Carboxylic acid group on the other side. So this is where it gets its name. It's got an amine group. It's got an acid group. It's called an amino acid. The 4th group is this R group, and that varies. So there are 20 different R groups that we encounter in nature. They're like, you can think of them as letters in an alphabet. So as we chain these amino acids together, we're going to stick another one here, right? And so on. As we continue to chain these on, we make combinations of these R groups. So one R group after another. And we can make a huge variety of molecules using this. One, we can vary their lengths. So some proteins are, 18, 20 amino acids long. Some are 2,000 amino acids long. It usually is somewhere in the order of hundreds. But we can make, at any position, we can put one of 20 different amino acids there. So we can make a huge variety of molecules here. We can do all kinds of craziness. And by tweaking those amino acids, we get different functions of the molecule. We can take something we had before, tweak it, and now it does something different, or maybe it does its old job better. And this is how these molecules get refined over generations, because the ones that do the job better, of course, are going to be passed on more often. We've talked about that plenty in natural selection. But you can make a huge variety of these. You don't have to know the structure of the amino acids. I did have a biochemistry class in college that I had to know the structure of every amino acid. In fact, I had to draw out proteins of specific sequences and draw every amino acid. So you might have to do that in college. And if you do, you should actually be happy about it, because memorization, it's cheap, man. It's super easy. It's not nearly as complicated as some of the questions they could ask you. So if you have to memorize the structure of the amino acids, you ought to be like, woohoo, memorization. Yes, please, more of that. Anyway, if you look at these, all of these are nonpolar, hydrophobic, right? And hopefully you know what that means, but there are no electronegative atoms on these, right? Or very few electronegative atoms compared to carbons and hydrogens, which have nonpolar bonds. So largely hydrophobic, right? These are ones that do not want to be next to water. You're going to have these amino acids on the inside of a protein. When a protein folds up, the part that's on the inside is going to be a bunch of hydrophobic amino acids that don't want to be next to the water on the outside. And so they end up huddled on the inside, leaving the amino acids that are hydrophilic, which are some of these, on the outside next to the water. So it helps give it its three-dimensional shape. It also allows the proteins to run through the membrane. Remember, that membrane has that hydrophobic barrier. If you have a spiral of protein running through a membrane, if the R groups all sticking on the outside are all hydrophobic, it's going to be perfectly happy in that membrane. And that's how we have membrane proteins. We have lots of them. So some are hydrophobic, some are hydrophilic. Some have charges. These have negative charges. These have positive charges. So they will interact with each other. The negatives like to be next to the positives. We get an ionic interaction when these guys start to fold up. So all kinds of things can happen. We even have some that have this sulfur. Cysteine has this sulfhydryl at the end, and it will form what's called a disulfide bridge. Basically, it's a covalent bond between two R groups. So as the protein folds up, it forms this covalent bond, and that's very strong. So it makes it really hard to unfold this protein. So you find these in places like, well, there are bacteria that live in hot springs. They use a lot of these because it helps keep their protein stable at high temperatures. It kind of holds it all together. We'll talk more about temperature and protein structure in a bit. So peptide bonds, of course, are formed the same way we've talked about other polymers being built. We use dehydration synthesis. Water comes out, right? And we form a bond at the place where those two electrons are dangling there, where we wanted to make those bonds. Where we took off the hydroxyl and the hydrogen, we got two places that want to bond. And so we formed that peptide bond there. And it's just called a peptide bond because of the structure here with the oxygen and the nitrogen. So when we build these proteins, we extend the C terminus. So what that means is, so I'm going to take this amino acid and I'm going to attach it to this one using dehydration synthesis. So I end up with this dipeptide. But if I were going to add another amino acid to this, I would only be able to add it to this end. I don't add to this end. So I always extend on the C terminus. This is ribosomes' job, right? Ribosomes build proteins, and they build them by extending the C terminus. They stick a new amino acid on that C terminus and keep building this train as it gets longer and longer and longer. They are adding amino acids to the C terminus. The N terminus, that's the beginning of the molecule. That's always the same. I shouldn't say it's always the same because sometimes we do chop it off and edit it. We do some things to change of amino acids after they're made before we get the final protein. There's a lot of little things that can tweak and be adjusted after the polypeptide has been assembled. So yeah, we say it's built into C, or we say it's extending the C terminus. And of course, like I said, this is ribosomes. Now, where ribosomes are is important because ribosomes do exist in the nucleus. They are actually built in the nucleus, but they aren't active in the nucleus. We only make proteins in the cytoplasm. So ribosomes in the cytoplasm. The ribosome is not a place. If you remember my poster at school, I have a poster that says, forbidden phrases in AP biology, and goes to the ribosome is one of those problems, right? One of those questionable phrases, because the ribosome isn't a place. There are jillions of them. You have thousands and thousands of these in each of your cells. And so going to a ribosome is not going to a place. That's like saying I'm going to a Michigander, to someone from Michigan. That's not a place. That's a thing that moves around. It can be different places. And ribosomes are like that too. So you go to the cytoplasm and encounter a ribosome. Precision of speech is important, especially in biology. So protein function depends entirely upon the protein shape. It's like a bird's wing, right? A bird's wing is shaped like that so the bird can fly. And the bird can fly because its wing is shaped like that. You can't really separate these two things. It's exactly true with these proteins. Hemoglobin carries oxygen very, very well. It picks up oxygen where it's needed to pick it up, and it dumps oxygen where it's needed to be dumped. It does that perfectly because of its shape. If you tweak its shape, problems are going to arise. You're going to have problems. Pepsin chops up proteins in your stomach. It works at low pH. It only does that because of its shape. Collagen is long fibers. If you can't construct collagen properly, you have scurvy. This is what happens with vitamin C. If you can't assemble collagen properly, you'll end up with scurvy. And that's, you need the enzyme that assembles the collagen protein requires vitamin C. We'll talk more about that later. But its shape is what gives it its function. It's long fibers in the case of collagen. So this is one of those fundamental, right? If you're building your web of biology, diversity is strength, right? The power that comes from diversity, from variation, that's one of the main cables that runs through your web. Another one, probably the other big, if you were to start the web with two huge cables that run through this, the other one is structure and function. That function is connected to structure on all levels. So diversity is strength, structure is function. Like that is the foundation of biology. So when we talk about protein structure, we have 4 levels in most proteins. Some only have 3, and I'll talk about that when we get to quaternary structure. But we start with primary structure. And the primary structure is simply the sequence of amino acids. Here's my N terminus. Here's my C terminus down here. And you can see I've got a lysine, a valine, a phenylalanine, a glycine, an arginine, cysteine, glutamic acid, leucine, alanine, methionine, lysine, arginine. That sequence of amino acids, I'm going to stop doing that. That sequence of amino acids is the primary structure. It's just the order of amino acids as assembled. You do need to know and understand each of these levels because AP does like to ask. They'll say, explain this in terms of secondary structure or something like that. Secondary structure is something that happens with small segments of the polypeptide that will form spirals and they will form beta-pleated sheets. So alpha helices, that's the spiral, and beta-pleated sheets, which is where the strands, it'll loop around and lay alongside itself, and it will make these, they look pleated, they look like they have folds in them because of the bond angles of the carbon chains. But these are all interactions between the backbones. So if I go back to this original amino acid we looked at, right, I'm going to extend off of here to make a polypeptide. So I've got this NCC, and then it'll be NCC, NCC. That's the backbone. So the backbone has these double bonded oxygens, and then it also has these nitrogens with hydrogens. And so we're able to do hydrogen bonding. So there'd be a nitrogen up here with a hydrogen, and it could do hydrogen bonding right here. This can happen if we're spiraling, then we end up with these 2 guys next to each other, or if we just turn around and like lay side by side, we can have these guys lining up next to each other. And they're equally spaced, so there'll be repeated units of it again and again where they can form multiple hydrogen bonds between these. And that's what you're seeing with this secondary structure. You can see there's a hydrogen bond here in this spiral. There's another hydrogen bond. There's hydrogen bonds all through this spiral in the alpha helix. And in the beta-pleated sheet, you can see the same thing, hydrogen bonds all through here, where that hydrogen off the nitrogen is hydrogen bonding with the oxygen that's stuck on that carbon, right? So that's a little delta minus there, a little delta plus there. That's that hydrogen bonding that's happening there. But these are backbone interactions. The R groups are not involved. In the case of beta-pleated sheets, the R groups are sticking out on one side. In the case of the alpha helix, the R groups are sticking out around the whole outside of this. So compared to covalent bonds, they're pretty weak. Compared to other intermolecular forces, they're pretty strong. But they form two different structures, alpha helices, beta-pleated sheets. Those are secondary structure. Backbone interactions, you need to make sure you know that too. I made it big and in red there. So again, this is secondary structure. Wow, that's really laggy. Tertiary structures when the R groups get involved. So the R groups have lots of different properties. Like I said, some are hydrophobic, some are hydrophilic, some can do hydrogen bonding, some can do ionic bonding, and some form those covalent disulfide bridges. All 4 of those things are shown here in this picture. These are what hold that folded up shape together. So here's hydrogen bonding between 2 amino acids that holds, this part to this part. Here's hydrophobic interactions that holds this part to this part, right? These are molecules not wanting to be next to water, like olive oil, right? Sticking side by side so that they can exclude water. Same as what happens in your cell membranes. The disulfide bridge we talked about, that's a covalent bond, very, very strong. Holds this strand to this strand in a very strong way. And then we've got that ionic bond that I talked about. Here's a negatively charged amino acid and a positively charged amino acid, and they're attracted to each other because of their opposite charges. It's an ionic bond. So all 4 of these things are what hold together that tertiary structure. That's the final folded up polypeptide. Now, when we get to quaternary structure, this is when multiple tertiaries, right, multiple folded up polypeptides, it can interact with each other, can stick to each other. And so that's what we're talking about with hemoglobin. We've got 4 different chains, right? We got 2 alphas and 2 betas. These are alphas down here, and these are betas. Chains of hemoglobin. Those 4 all come together to make a proper, fully functioning hemoglobin. Collagen's even a little more complicated than that. It kind of polymerizes and makes multiple strands that weave together. But that's quaternary structure. And a lot of proteins don't have a quaternary structure. Insulin, for example, is a protein hormone. It's a small molecule. It's just a single polypeptide folded up. So it only has tertiary structure. It doesn't have a quaternary structure. So here's all 4 of those levels. And the main thing you need to know about them on one slide, which is nice. Of course, the primary sequence is determined by the DNA. That's that central dogma that you may or may not remember. We take DNA, we use it to make an RNA copy, and that RNA copy is used to make proteins, is used as the recipe by the ribosome to tell us the order of amino acids. And so that came from the DNA. That's the thing you inherit, is the recipes for those amino acid chains. It's the sequences of amino acids. That's what you got from your parents. And with only a single nucleotide change in the DNA, we can change a single amino acid. And it may not seem like a big deal, but we're changing the primary structure. So we go from a glutamic acid here to a valine here. And in the case of sickle cell anemia, right, the glutamic acid is hydrophilic. It's got a negative charge because it usually gives that proton, and body pH is going to let go of that hydrogen. And so it's got a negative charge, and it's very happy next to water. So that's part of what makes hemoglobin a nice ball, right? It's a ball-shaped protein. That's the globin part. So when you got a red blood cell, so let's say I got a red blood cell and it's filled with hemoglobin, these little balls. It's jiggly and it's bouncy. It's kind of a flattened disc. It's kind of biconcave is what they call it. It's kind of indented here. You can see it on the left. Looks like donuts, but the holes don't go all the way through. But so those are, bounce around and squish around and go through blood vessels and capillaries, even capillaries that are so small that they have to go through single file. They can do all that. But if you take the hemoglobin and you change on that beta chain, you change one glutamic acid to one valine. This spot is hydrophobic and does not want to be next to water. Now it's on the outside of the protein. It's supposed to be, hydrophilic. Everything on the outside should be happy next to water, but this guy doesn't want to be next to water. So this guy on one hemoglobin and that same guy on another hemoglobin can exclude water by getting together. But remember, there's two places where this happens because there's two beta chains here. So it'll stick one, and then it'll stick another, and then it'll stick another on that, and another on that, and another on that. And now we've got chains of hemoglobin. Instead of a bag of little balls, you've got a bag of spears. And that makes the cells really irregularly shaped. We usually call them sickle cell because they look kind of sickle shaped. But that one looks like a cruller. That one looks like a slipper. That looks like a cat kind of going that way. They get really irregularly shaped. And so when they go in those capillaries, they can block it up and jam it up and block blood flow to whatever is over here. If that's your brain, you're having a stroke. If that's your heart, you're having a heart attack. If that's any other tissue, it's likely to die. What we have there is one amino acid, a problem with the primary structure that then gets passed on to the quaternary structure. It becomes a problem for the quaternary structure because now instead of just being a tetramer of 4 parts, it's a chain of tetramers, so it becomes this really huge molecule. So it makes a new quaternary structure that's really problematic. So hopefully that all makes sense in the level of structure. So you can see that primary is a problem, quaternary is a problem there too. So we also have this idea of protein denaturation. And this is the unfolding of the protein. So I want you to try and answer these questions. Think about the structure of protein and answer these 3 questions. So pause the video and answer these. And talk to a buddy if you can. So I added those little pause the video things. I'm not sure if I'm going to keep doing that. I'd probably just use the same one over and over again. That might be fun. Thanks, Bootsy Collins, for the bass melody, by the way. So when you're looking at these R group interactions, right, covalent bonds, you can't break those with just heating, by heating things up, right? I can boil water and not break hydrogen-oxygen bonds, no problem. But I will disrupt the hydrogen bonds between the water, and that's what allows the water to boil away. I can also disrupt hydrophobic interactions with heating. I can disrupt ionic bonds with heating. I can get those things moving apart, especially in solution like this. So I'm going to disrupt all 3 of these first ones with heat. And so I'm going to lose all 3 of those, but not covalent bonds. So primary structure, is that going to be affected by heating? No, that relies entirely on covalent bonds, right? Secondary structure, that's hydrogen bonding. We're losing secondary structure because we're going to break hydrogen bonds with heat. Tertiary structure. Yeah, absolutely. All 3 of those first 3 are in tertiary structure, determine the tertiary structure. So it's going to screw that up. If we got a lot of covalent bonds, that'll help, but only so far because we're going to disrupt all the rest. And quaternary structure, that's usually hydrophobic interactions. That's polypeptides sticking to each other with hydrophobic interactions. So yes, also easily disrupted. So what we're going to have is this, clump of chains all folded up. We heat it and it becomes just noodles. It's just a mess. So long strands. So what's that do to the function of the protein? Remember that the function of the protein is entirely dependent on the structure of the protein. We just turned it into a bowl of noodles. It's not going to do what it's supposed to do anymore. You've actually seen denaturation. If you've ever fried an egg, here's my breakfast this morning. The whites of the eggs it's a protein called albumin suspended in water. It's largely this protein. And this protein is just a ball shape, and it kind of helps give the water a certain osmolarity, and it gives it some thickness, and it's, cushioning for the growing embryo inside the egg. It also provides water and some protein later on in development. But if I put that egg in heat, I'm going to lose the hydrophobic interactions, the hydrogen bonds, and the ionic interactions. So all that albumin, that is this nice ball-shaped, floaty, hydrophilic outside, is now going to unravel. And when it unravels, all those hydrophobic R groups are now exposed. It's precipitating out of solution. It's not wanting to dissolve anymore because it's hydrophobic. So much hydrophobic is exposed. And so it precipitates out. You can no longer see through it. It becomes white. Right? And that's what happens when solutions precipitate. If you've ever seen a precipitation reaction, you see some white powder come out oftentimes. That precipitation, that's you being able to see it because it's no longer in solution. It also gets tough, right? And that's because those strands are long fibers and they're laying every which way and they've got water trapped in between them. So they're tough and still kind of jiggly because, like Jell-O, which is the same thing, it's collagen with water trapped in. It's long strands with water trapped in between. It's just that same type of texture, right? And so it's just because those proteins have unraveled when you fry an egg. You can actually denature proteins in other ways too. Excessive amounts of salt can do it because that's going to interfere with ionic bonds and hydrogen bonds, anything with a charge, because when you put salt in there, right, that's going to be attracted to anything with a charge. So a lot of salt can disrupt this. Acids can do this. Definitely, you can change these interactions. You're going to put protons in some of these positions instead of each other, and so that disrupts a lot of things. Actually, I don't know if you guys remember what vines are. You probably do just because of Vine compilations. But here's a vine that I made a long time ago, denaturing a protein, denaturing the albumin in egg whites with vinegar. So that's kind of fast. I ran it through a few times so you could see, but if you go back and look at it, I add vinegar to an egg, and then later when I look at it, it's cloudy. And that's because the white of the egg has started to cook. It started to denature. It's not actually cooking or anything. And then I added some sriracha just for flavor, and then I drank it, and I'm not really sure why. So anyway, that's proteins and protein structure. Next lecture will be enzymes, which are just proteins that catalyze chemical reactions. So we'll talk a lot about that in the next lecture. You guys are doing great. Hang in there.